Good morning, everyone. Today we are going to talk about the physiology of the large intestine, and we are going to mainly focus on the secretory and absorptive activities of the large intestine, as well as the main motility patterns of the large intestine, including the defecation reflex. The objectives are to appreciate that the mucosa of the colon has no villi and it secretes no enzymes, and to recognize the active production of mucus and bicarbonate by the mucosal cells and its relevance, explain the functions of the colon, appreciate the mass movements that occur in the colon and their control, and describe the defecation reflex. And very important point is to know the functional importance of the colonic or large intestinal microbiota or the bacterial flora, and to list the motility disorders of the colon and name the two conditions that constitute the inflammatory bowel disease. Here, as you remember, that the large intestine is the terminal part of the alimentary canal. It is the terminal portion of the GIT, and it extends from the ileum to the anus, from the ileum at the ileocecal valve, and it ends in the anus, which is the opening of the terminal portion of the alimentary canal to the exterior. It has a length of 1.5 meter, which is shorter than that of the small intestine that we previously studied in one of the previous lectures. Structurally, it is divided into four major parts, and these parts, as you remember from the anatomy, the cecum, and then we have the colon, and then we have the rectum and the anal canal. As I said, it opens to the exterior through the anus. The colon has four major portions. It has an ascending part, transverse part, descending part, and the sigmoid-shaped sigmoid colon. So the four major regions, as I said, are the cecum, the colon with its ascending, transverse, descending, and sigmoid portions, the rectum, and finally the anal canal. The wall of the large intestine is composed of the four basic layers as the other parts or segments of the alimentary canal. These are the mucosa, submucosa, muscularis externa, and the serosa. In terms of mucosa, the mucosa of the large intestine, as you can see, it is composed, the surface mucosa composed of two main cells, the absorptive cells. These are the majority of cells, and we have the goblet cells. So it is composed of simple columnar epithelium that contains the absorptive cells. Their function is mainly the absorption of water and electrolytes, and the goblet cells that secrete mucus for lubrication as other parts of the alimentary canal. The mucosa also has a lamina propria like other segments of the alimentary canal, which is loose connective tissue. Then we have the muscularis mucosa, this part here that separates the mucosa from the submucosa. The large intestine, similar to the small intestine, it has crypts of Lieberkühn or large intestinal glands. They are long, straight, tubular intestinal or colonic glands that extend the full thickness of the mucosa. They are longer than the crypts of Lieberkühn that are present in the mucosa of the small intestine. Please notice that the large intestine has no villi, so there are no plica circularis and there are no villi, but the surface absorptive cells, here the enterocytes of the large intestine, have microvilli, and these are finger-like projections of the apical membrane, which is the phospholipid bilayer of the absorptive cell. And the presence of microvilli will give me a hint that they have an important absorptive role. It is meant to increase the surface area of the absorptive mucosa of the large intestine. Here in this illustration, as you can see, we have the small intestine, or a piece of the small intestine wall, and a piece or portion of the large intestinal wall. And you can see the difference in mucosa, where we can see here in the small intestine the presence of villi and the shallow or short crypts of Lieberkühn. In the large intestine we have deeper colonic glands or crypts of Lieberkühn, and there are no villi on the surface mucosa. So the crypts of Lieberkühn, or the colonic glands, these are deeper than those in the small intestine. They have a depth of 0.4 to 0.6 millimeter. They are lined with different types of cells. We know some of them from the previous lectures. Here in the large intestine, they are lined with absorptive cells, which is opposite to the small intestinal crypts of Lieberkühn. In the crypts of Lieberkühn of the small intestine, we have secretory cells that produce the intestinal juice. So in the large intestine, the crypts are lined mainly by absorptive cells. These are called absorptive cells or absorptive enterocytes. They absorb water, sodium chloride, and they secrete potassium and bicarbonate. But their main function is absorption. That's why we call them absorptive cells. The absorbed water contributes to the maintenance of water balance by the body. The goblet cells, which is the second type of cells, they are less abundant than the absorptive cells. They secrete mucus for the lubrication, and they provide an adherent medium for holding the fecal material together. Then, as other glands in the alimentary canal, like the gastric glands as well as the small intestinal glands, there are stem cells that will give rise to the other cells, mainly the lost epithelial cells that are shed regularly due to the tear and wear phenomenon. Finally, we have the enteroendocrine cells, and we have the L cells that secrete peptide YY. This is a hormone that decreases appetite. We have glucagon-like peptide 1, which stimulates insulin release, and this will reduce blood glucose level. And we have also the glucagon-like peptide 2, which promotes the growth of the large intestinal mucosa and also helps in the function of the gut. Okay, now what are the functions of the large intestine? The overall functions of the large intestine are to absorb salt and water to produce semi-solid or solid fecal material, and absorption of water and salt is important for the water and electrolyte balance. Also to absorb short-chain fatty acids produced by the colonic bacteria or the intestinal flora, and also they are called the gut microbiota, and we'll talk about them in one of the slides at the end of the lecture. So they are produced from non-absorbed carbohydrates that enter the colon. The third function is the production of certain vitamins by the colonic bacteria and their absorption, and these vitamins are very important for the homeostasis in the body, and we'll cover them when we'll talk about the microbiota. Number 4 is the formation and storage of feces or fecal material, and the expulsion of the fecal material from the body in the defecation reflex. Therefore, the large intestine functions as two organs and can be subdivided into two parts from the above mentioned functions. We have an absorptive colon, which is function 1 and 2, and this is usually the proximal half of the large intestine, and we are mainly talking about ascending and transverse colons. It is the site for the bacterial production of fatty acids and their absorption, together with most of water and electrolytes. So this part usually covers function 1, 2, and 3. And we have another part of the large intestine, which is called the storage colon, which is the distal half, and this is the descending and sigmoid colon. The distal half functions principally for the feces storage until conditions are suitable for defecation. Now we'll talk about the absorption of the large intestine. As you can see that every day what enters, or what are the fluids that enter the alimentary canal and the fluids that leave the alimentary canal. First we see that we produce about 1 liter of saliva. We ingest about 2.3 liters of liquid, either as drinks or with the food. We have 2 liters of gastric juice, 1 liter of bile, and 2 liters of pancreatic juice, and the intestine or the small intestine produces about 1 liter. So totally 9.3 liters will pass through the alimentary canal every day. The majority of this fluid is absorbed in the small intestine, which accounts for 8.3 liters. So we have about 1 liter that will leave the small intestine as chyme through the ileocecal valve to enter into the large intestine. The large intestine will absorb about 90% of that, which is 0.9 liters, a bit less than 1 liter. So totally what is absorbed from the alimentary canal is 9.2, and we are left with 100 milliliter or 0.1 liter, which is the fluid that is lost in the fecal material or with feces. So every day the colon receives 1 to 1.5 liters of chyme from the small intestine through the ileocecal valve. The chyme delivered to the colon is composed mainly indigestible food residues, and this is cellulose, and unabsorbed biliary components, unabsorbed fluid and electrolytes. And then the colon will absorb most of the water, about 900 milliliter or 0.9 liter, as I said, and electrolytes, mainly sodium, chloride, calcium, magnesium, and zinc. We said the colon secretes potassium and bicarbonate, but the ions that are absorbed are sodium, chloride, and the divalent cations. Divalent cations like calcium, magnesium, and zinc. And the absorption of divalent cations requires a slightly acidic environment, and we know that the pH in the colon is slightly alkaline, so we'll see how the microbiota can help in the absorption of these divalent cations later on. What remains out of the chyme, or what remains after the absorption of water and electrolytes and the short chain fatty acids and the bile acids, will go or will be eliminated as feces. Please note that cellulose and other indigestible substances are important in our diet. Though we don't digest them, they provide bulk and they help maintain regular bowel movements, and we'll see that cellulose is important also to maintain the gut microbiota, the colonic bacteria. The maximal absorptive capacity, as you can see here, the maximal absorptive capacity of the large intestine mucosa is 800 ml. So it can absorb up to 8 liter. Usually at rest or in normal conditions, it absorbs about less than 1 liter, but it can even deal with 8 liters. So usually diarrhea won't happen until the amount of fluid that is delivered to the large intestine is more than 8 liter, or its absorptive capacity is decreased by any disease that damages the large intestine mucosa. So what are the secretions of the large intestine? Can the large intestine produce any secretion, and does this secretion contain any enzymes? Let's see. The secretion of the large intestine is much less than that of the small intestine. It is very small amount of secretion, and it's mainly of mucus. The colonic secretion is composed of mucus to protect the mucosa against chemical and mechanical injuries, and to provide lubrication to facilitate the passage of feces. The other secretion is bicarbonate that will neutralize the irritating acids produced by the gut microbiota. Gut microbiota will undergo fermentation, and this fermentation will produce acids, and these acids are irritating. So the acids will help in the absorption of divalent cations, but they are quickly neutralized by the secreted bicarbonate. Another secretion is potassium via potassium channels present in the apical membrane of the lining enterocytes, and this secretion is through active transport, and it is under the control of aldosterone hormone. There is an example of diarrhea caused by excessive secretion, which is the cholera enterotoxin that stimulates potassium secretion, and this will lead to diarrhea characterized by excessive loss or fecal loss of potassium. Secretion increases in response to direct mechanical and chemical stimulation of the colonic mucosa by local, short reflexes and long central parasympathetic reflexes, similar to other segments of the alimentary canal. The large intestine does not secrete any digestive enzymes. Please remember this. No digestive enzymes are secreted or are part of the large intestine juice. And there are no brush border enzymes on the apical membrane of the absorptive cells. So the apical membrane does not contain any brush border enzymes, which is opposite to the small intestine. The only digestion that happens in the large intestine happens in the gut microbiota, okay, the flora or the bacteria that are present in the lumen. Therefore, no digestion occurs in the large intestine except the digestion of some cellulose by the gut microbiota or the colonic bacteria. And this digestion provides nutrients for their own metabolism. So this is for the own benefit or the benefit of the microbiota. Intense irritation of the colon, especially the colonic mucosa, can result in excessive secretion of water and electrolytes, and this will lead to diarrhea as a protective mechanism. So whenever a segment of the large intestine becomes intensely irritated, example by bacterial infection during enteritis, the mucosa secretes extra large quantities of water and electrolytes, including potassium, in addition to the normal viscid alkaline mucus. This acts to dilute the irritating factors and to cause rapid movement of the feces towards the anus, and the result is diarrhea with loss of large quantities of water and electrolytes. This is harmful to the body, but on the other hand, the diarrhea also washes away the irritant factors, and this will promote earlier recovery from the disease. Now let's talk about the motility of the large intestine after finishing the secretion and absorption that happen in the large intestine. So what are the main motility patterns, and whether they resemble or they are different from those that happen in the esophagus, stomach, and the small intestine, and whether we have a certain or special motility patterns that are characteristic of the digestive period, and whether we have motility patterns that happen during the interdigestive state. Let's see that. So the features of the large intestinal motility: the movements of the colon are normally sluggish. We don't need fast transit of fecal material or of the chyme at the beginning of the large intestine. So we need slow movement. Intense colonic movements are not required for its function because we want to absorb water and electrolyte, the vitamins produced by the microbiota, and the short chain fatty acid. So we need some time for the absorption and for the action of the microbiota. Though sluggish, the movements have characteristics similar to those of the small intestine and can be divided into mixing movements, and we call them haustrations or haustral contractions. These are slow movements that promote absorption. So they are just mixing movements that are allowing the absorption of the content. Propulsive movements, we call them in the large intestine mass movements that eventually lead to defecation when the conditions are suitable. The pattern of colonic motility is similar in both the fed and interdigestive states. So the pattern of motility is the same. So we have mixing movements and mass movements during fed state and during the interdigestive states. We don't have migratory motility complex like the stomach and the small intestine. So please pay attention to that. The movements are the same whether we are during the interdigestive state or during the fed state. Here we have two motility patterns, the segmenting contractions, and these are the mixing movements. So they happen everywhere along the large intestine, but mainly in the proximal part or the proximal half. So the segmenting contractions happen mainly in the ascending and transverse colon, or the first half of the large intestine. The mass movements, the second motility pattern, we can see it happens mainly in the distal half of the large intestine, and usually they lead to the initiation of the defecation reflex, because they will push the fecal material into the rectum, and once the rectum is distended, defecation reflex will be stimulated or initiated. So as you see, it happens throughout the day, the mixing movements. The mass movements, they happen 3 to 10 times per day, and usually they succeed in initiating the defecation reflex 1 to 3 times per day, as we'll see in the defecation reflex. Haustrations or haustral contractions are the first motility pattern of the large intestine. These are slow sequential circular muscle contractions that are combined with contraction of the tinea coli, the 3 bands of longitudinal muscles that are present in the or are part of the muscularis externa layer of the wall of the large intestine. The unstimulated portions of the large intestine will bulge outward into bag-like sacs called haustri. And this is what you studied in the anatomy as a morphological feature of the large intestine. Each haustration or each haustral contraction usually reaches a peak intensity in about 30 seconds and then disappears during the next 30 seconds. So 30 seconds of contraction followed by 60 seconds or 1 minute of relaxation. After a few minutes, and this usually 30 minutes, new haustral contractions occur in nearby previously relaxed area. So every 30 minutes, certain areas will undergo 30 seconds of contraction and 60 seconds of relaxation. Then after half an hour, the areas that were inactive will start the haustral contractions with contractions of 30 seconds followed by relaxations of 60 seconds. So new haustral contractions occur nearby previously relaxed areas. This will slowly mix and propel the colonic contents towards the anal canal. In this way, all fecal material is gradually exposed to the mucosal surface of the large intestine, and fluid as well as dissolved substances are progressively absorbed until only about 1 milliliter of feces are expelled each day. So about 0.9 liter will be absorbed, and 0.1, with the average between 50 to 150 ml of feces, will be finally expelled from the body. Haustration depends on the activity of the interstitial cells of Cajal. They give rise to slow wave activities. We already know that from the stomach and small intestine lectures. This will lead to contractions that are triggered by distension of the colon and the action of both intrinsic and extrinsic nerves. Now let's talk about the second type of movement, which is the mass movements. Much of the propulsion in the cecum and ascending colon results from the slow but persistent haustral contractions. So the propulsion in the first part of the large intestine is through the haustral contractions that transform the semi-fluid chyme into semi-solid fecal material due to the absorption of water and electrolytes. And this usually requires about 8 to 15 hours to move chyme from the ileocecal valve through the colon. Mass movements take over the propulsive role, and they happen 1 to 3 times per day, and they rapidly propel colonic contents from the cecum to the sigmoid colon. Each time, these movements usually last for a few minutes, for about 15 minutes, especially during the first hour after eating breakfast. So those mass movements that happen 1 or 3 times, or in some people up to 10 times, they last only for 15 minutes. The mass movement is a modified type of peristalsis characterized by the following sequence of events. Mass peristalsis starts as a constrictive ring. So circular muscles will contract, and this usually starts or happens in the transverse colon. The colonic segment, about 20 centimeter length, distal to the constrictive ring, loses its haustration and contracts as one unit. So you have a region of the large intestine that is contracting as one unit, and it has a length of 20 centimeter. This will propel the fecal material in an anal direction. So constrictive ring followed by loss of the haustry distal to that constrictive ring, and then the area with lost haustry will contract as one unit, and this will propel the fecal material towards the anus. The force of contraction becomes progressively stronger for about 30 seconds, and relaxation occurs during the next 2 to 3 minutes. Then another mass movement occurs in a more distal segment, and this process will keep repeating for 15 minutes, which is the entire period of one mass movement. Series of mass movements usually persist for 10 to 30 minutes, on average 15 minutes, as I said, and this will push the fecal materials toward the anus. Then they cease and reoccur every 8 to 24 hours. So they stop after 15 minutes or maximum 30 minutes, and they will repeat every 8 hours or every 24 hours. If mass movements force a mass of feces or fecal material into the rectum, distension of the rectum will bring the desire for defecation, and this will initiate the defecation reflex. Mass movement can be initiated or is initiated by gastrocolic and duodenocolic reflexes. What do we mean by gastrocolic and duodenocolic reflexes? Distension of the stomach will initiate or stimulate mass movements in the colon, and the same distension of the duodenum by chyme emptied from the stomach can also initiate or stimulate the mass movements in the large intestine. So appearance of mass movements after meals is facilitated by gastrocolic and duodenocolic reflexes. These reflexes result from mechanical or chemical stimulation of the mucosa of the stomach, of the stomach and duodenum wall. Mucosa irritation and the stretch stimulates the entire wall thickness. They are mediated, or both reflexes are mediated by autonomic nervous system. These are long central parasympathetic reflexes, and they are lost when the extrinsic autonomic nerves to the colon have been removed or cut during surgery. And then also these reflexes are mediated by GIT hormones, mainly gastrin and cholecystokinin. Gastrin, that is released mainly from the stomach, cholecystokinin mainly from the intestinal glands of the small intestine. Irritation in the colon can also initiate intense mass movements independent of these two reflexes. Example, a person who has an ulcerated condition of the colon mucosa, like in condition called ulcerative colitis. This will lead to frequent mass movements that persist almost all the time. So the patient is having frequent pain and also diarrhea. Now let's talk about the defecation reflex. I said defecation reflex is initiated by pushing fecal material by mass movements into the rectum. Distension of the rectum will initiate the defecation reflex. So during most of the day, the upper portion of the rectum generates segmental activity which tends to prevent emptying of the colonic contents into the rectum. These movements ensure that the rectum is usually empty. When a mass movement forces feces into the rectum, distension of the rectal wall will lead to the stimulation of stretch receptors that are part of the wall. And this will initiate defecation, or will initiate defecation reflexes that empty the rectum, desire for defecation. Please note that I said defecation reflexes. So in introduction you studied one reflex, but here at higher level we talk about at least two reflexes, and we'll talk about them in the coming slide, or here in this slide. So these two defecation reflexes are intrinsic defecation reflex, which is relatively weak. It is a local reflex that happens at the level of the wall of the large intestine. It is mediated by the local enteric nervous system in the rectal wall. Again, the intrinsic or local defecation reflex is relatively weak, and it needs the support of a stronger defecation reflex to ensure defecation. And this is the parasympathetic defecation reflex, which is strong reflex mediated by the sacral segments S2, S3, S4 of the spinal cord. It is a central reflex. It fortifies the relatively weak intrinsic defecation reflex to be effective in causing defecation. As you can see here in this illustration, we have this or these components of the defecation reflex. This is the rectum, we have the anal canal, and we have the internal and external anal sphincters. Stretch of the rectum by fecal material will stimulate mechanoreceptors in the wall of the rectum. This will send information to the sacral segment of the spinal cord, S2, S3, and S4, and will lead to the processing of the information at the level of the spinal cord, and finally will result in impulses to the wall of the spinal cord, to the wall of the large intestine, or to the wall of the rectum. And this is in terms of contraction of the muscles or circular muscles in the parts proximal to the fecal material, and relaxation of the internal anal sphincter, which is under involuntary control. If conditions are suitable, pathways from the cerebral cortex will mediate the relaxation of the external anal sphincter, and defecation will happen. So defecation reflexes: the intrinsic defecation reflex, the local defecation reflex, which is a weak reflex Distension of the rectal wall by feces will initiate afferent signals that spread through the myenteric plexus to initiate peristaltic waves in the descending colon, sigmoid colon, and rectum. So the signals are carried through the myenteric plexus to more proximal parts of the large intestine. These segments will contract, and this will force fecal material towards the anus. As the peristaltic wave approaches the anus, the internal anal sphincter is relaxed by inhibitory signals from the myenteric plexus. If the external anal sphincter is also consciously or voluntarily relaxed, at the same time defecation will occur. In terms of the strong parasympathetic defecation reflex that is essential for defecation, stimulation of nerve endings in the rectum that express the stretch receptors will lead to signals that are transmitted first into the spinal cord and then reflexly back to the descending colon, sigmoid colon, rectum, and anus by way of parasympathetic nerve fibers in the pelvic nerves. Parasympathetic signals greatly intensify the peristaltic waves and relax the internal anal sphincter, and this will lead to the conversion of the intrinsic myenteric defecation reflex from weak effort into a powerful process of defecation. So without the parasympathetic defecation reflex, defecation won't happen. So defecation signals enter the spinal cord also initiate other effects. These signals, when they enter the spinal cord, they will initiate other effects. One of them is taking a deep breath that is followed by expiration or exhalation against the closed glottis. This is called the Valsalva maneuver with contraction of the abdominal muscles. So this will increase the intra-abdominal pressure. This will put pressure from the outside on the colon, especially the rectum and anal canal, and this will aid in the expulsion of feces. So an external pressure on the rectum will help in evacuation of the content of the rectum. If the circumstances are inappropriate, if the conditions do not allow defecation to happen, what will happen? The act of defecation will be postponed. The internal anal sphincter contracts and the rectal contents are returned to the colon by retroperistalsis. If we want to stop the desire for defecation, we should empty the rectum. How this can be achieved if defecation or conditions are not suitable by defecation? We return the content of the rectum back to the sigmoid colon. How this can be achieved? This is achieved by retroperistalsis, which is peristalsis in the opposite direction, okay, from the rectum to the sigmoid colon. So returning the content to the sigmoid colon until the next wave of mass peristalsis will bring those fecal material back to the rectum. So if we empty the rectum, the sensation to defecate will disappear. The external anal sphincter is under voluntary control. That's why we can control, or adults can control their defecation reflex, and if it is voluntarily contracted, internal anal sphincter also will contract, and the fecal material will be returned to the sigmoid colon by retroperistalsis. Now, what is the composition of feces or of the fecal material? Of course, it contains water, about 100 ml of water, and this represents about 75 of the fecal mass, and 25% is in the form of solid matter. Fecal material has a pH between 7 to 7.5. It is slightly alkaline. The solid matter that represents about 25% of the fecal material mass is composed of 30% dead bacteria, including the gut microbiota, because these are living organisms, they can die, they reproduce, so 30% is in the form of bacteria. 10 to 20% in the form of fat, 10 to 20% as inorganic matter, 2 to 3% proteins, and 30% undigested roughage from the food and dried constituents of the digestive juices such as bile pigments and the sloughed epithelial cells. We know that the alimentary canal throughout its length has stem cells, and the stem cells will divide by mitosis to produce new epithelial cells, so the old cells will be sloughed and lost in the fecal material. The brown color of the fecal material is caused by stercobilin and urobilin, and these are derivatives of bilirubin. You should study them in biochemistry, I guess, and also in the blood lecture in the introduction course. The odor of the feces is caused principally by products of the gut microbiota action, and these products are indole, scatol, mercaptans, and hydrogen sulfide. Now let's talk about the gut microbiota, or the colonic bacteria, the beneficial bacteria, or the friendly bacteria that we have in our large intestine. Numerous bacteria, especially bacilli, are present normally in the large intestine, especially the absorbing colon, so the proximal half. This bacterial population of the large intestine is called the intestinal flora, the colonic bacteria, or the gut microbiota. So all have the same meaning. The total number of bacteria in the intestinal flora is about 300 trillion, and this number is about 10 times the total number of the cells in the human body. So we have more bacteria in our large intestine than our total body cells, or the number of our total body cells. They are capable of digesting small amounts of cellulose that human being cannot digest and absorb. So they provide by this process few calories for their own metabolic use. They are harmless, and in fact they provide beneficial functions. So what are these functions? They synthesize or produce vitamins, especially vitamin K, vitamin B12, thiamine, riboflavin. So they have a great nutritional contribution to us. Our daily intake of vitamin K is not sufficient for the production of the clotting factors. So we need that vitamin K that is produced by the bacterial or by the colonic bacteria or the microbiota. And people who are on for long period or on antibiotics that disturb or kill the colonic bacteria usually have problem of vitamin K deficiency in addition to the other vitamins. And this shows us how much important the contribution of the colonic bacteria in providing our daily requirement of vitamin K, especially vitamin K. The bacteria formed vitamin K is especially important because the amount of this vitamin in daily ingested food is normally insufficient to maintain adequate blood coagulation. The other function of the colonic bacteria is they raise colonic acidity because they produce acids during fermentation. And as I said, the absorption of divalent cations like calcium, magnesium, and zinc, they require slightly acidic environment close to the mucosal surface. And this is achieved by the acids produced by the colonic bacteria during fermentation process. The third nutritional contribution, they enhance intestinal immunity by competing with potentially pathogenic microbes for nutrients and space. So if we use antibiotics, the normal flora will disappear and they will be replaced by pathogenic bacteria, and then we'll got more problems. So they compete with pathogenic microbes, for space, as well as nutrients. The colonic bacteria also produce various gases during fermentation process, and these are carbon dioxide, hydrogen gas, and methane. Most of these gases will be absorbed through the mucosa, and the remainder, or what remains, will be expelled as flatus. Okay. Finally, let's mention some motility disorders of the colon that you'll study them more in the integrated phases, integrated modules, as well as in phase 3 in medicine, inshallah. So one or the first motility pattern, which is a common disorder, is constipation. Opposite to that is diarrhea. We have a condition called irritable bowel syndrome, and finally the inflammatory bowel diseases, and these are two main entities: Crohn's disease and ulcerative colitis. You will study them, I guess, in some of the pathology lectures and mainly in the medicine part of phase 3. And here it is a summary of the digestive activities that happen in the large intestine. In the lumen, the bacterial activity breaks down undigested carbohydrates, proteins, and amino acids into products that can be expelled in feces or absorbed and detoxified by the liver, and also the synthesis of certain vitamin B vitamins and vitamin K. The mucosa secretes mucus that lubricates the colon, protects the mucosa, and holds the fecal material together. And also the mucosa undergoes absorption, mainly water absorption. This will solidify fecal material and contributes to body water balance. Solutes are also absorbed, including ions and some vitamins. The muscularis, muscle layer, Haustral contractions, peristalsis, mass peristalsis, and defecation reflex. Please remember that at the end of this lecture that the small intestine is the major part of the alimentary canal, or is the main site for water absorption. Okay, usually a question that comes in exam: what is the main part for water absorption in the alimentary canal? And most students will tick the large intestine. No, we saw that 8.3 liters are absorbed in the small intestine, and the remaining 1 liter that will be delivered to the large intestine, just 0.9 liters are absorbed. But if I ask you what is the main function of the large intestine, you can say water absorption. Most or the majority of water in the alimentary canal is absorbed in the small intestine, but the major function of the large intestine is water absorption. Please remember this comment, and thank you for your patience. And with this slide we end our lecture. These are the references that were used to prepare this lecture. Thank you very much. Please stay safe.